This lecture covers the structure and function of the plasma membrane, also known as the cell membrane. This is a drawing of the plasma membrane. It shows the phospholipid bilayer. That means two layers of phospholipid molecules, and proteins that go through this phospholipid bilayer, or integral proteins, and proteins that sit on the outside of this phospholipid bilayer, known as peripheral proteins. This is another drawing of the plasma membrane. In the following slide, we're going to take a look at one of these phospholipid molecules. This is a single phospholipid molecule. You saw a number of these molecules in the previous slide. They actually formed two layers in the cell membrane. Phospholipid molecules are composed of two parts. One is the head, which contains a phosphate. It is hydrophilic. That means it loves to be in contact with water. The second part is the tail. It contains two fatty acids, and because they are fatty in nature, they are hydrophobic, or they fear water. They do not want to be turned toward water. The cell membrane, also known as the plasma membrane, is a biological membrane that separates the interior of all cells from the outside environment. The cell membrane is selectively permeable to ions and organic molecules and controls the movement of substances into and out of the cells. The basic function of the cell membrane is to protect the cell from its surroundings. In order to understand the plasma membrane and how it works, there are certain basic processes that you need to understand. One of those is the process of diffusion. Diffusion means the movement of a substance from an area of high concentration to an area of lower concentration. You can see this illustrated in the diagram below. Although diffusion is the movement of a substance from an area of high concentration to one of lower concentration, there is a minimal amount of backward movement. So what we want to look at is the net flow, or the difference between what moves from high to low concentration and what's moving back to high concentration. That's known as net flow, and you can see it in the diagram below. Substances can pass through membranes in a number of ways. One is osmosis, which is a passive process, meaning it does not require energy. Active transport is another way, but it does require energy and therefore is said to be active. Phagocytosis, which is also an active process, and pinocytosis, an active process, meaning it requires energy. We're going to look at each of these in the following slides. The first process is osmosis, a passive process. It's the movement of water across a membrane from an area of higher concentration to an area of lower concentration. You can see that in the diagram on the right. Only the water molecule is small enough to move through the holes in the membrane. The second way substances can cross a membrane is by active transport, which requires energy. Active transport is the movement of a substance across a membrane from an area of lower concentration to an area of higher concentration. Because you're moving against a gradient, energy will be required, and that energy is ATP. Phagocytosis is another way substances can cross a membrane. Phagocytosis is the movement of a large particle across a membrane without moving through the pores or through the holes. It is an active, energy-requiring process. Click on this link to view an animation of phagocytosis occurring. On the following slide, you can watch a video. You can see the process of phagocytosis in the diagram below. On the left is a blood neutrophil, which is a white blood cell. Its job is defense. On the right, you can see that neutrophil engulfing a pathogen, perhaps a bacterium, and taking it in through the process of phagocytosis. Pinocytosis, another active or energy-requiring process, is identical to phagocytosis, except it is the movement of a thick viscous liquid and small particles across a membrane instead of a large particle. You can see the process of pinocytosis in the diagram on the right. I'm sure you've heard the term solution before. A solution is a solid dissolved in a liquid. That's sometimes described as a solute plus a solvent equals a solution. The solute is the solid and the solvent is the liquid. There are three types of solutions: a hypertonic solution, a hypotonic solution, and an isotonic solution. We'll talk about each of these on the slides that follow. First, the hypertonic solution. Hyper means greater than or more than. So a hypertonic solution is a solution that is more concentrated than the cells. If you look at the diagram on the right, you can see that the concentration outside the cell is much greater than the concentration of particles inside the cell. So that means that the concentration of water inside the cell is greater than the concentration of water outside the cell. Since the pores in the cell are very small, only water can move, and it will move from an area of higher concentration to an area of lower concentration, or from inside the cell to outside the cell, and that causes the cell to shrink. So let's recap the previous slide. In a hypertonic solution, only water can move, and it's because the pores are small and water is the only thing small enough to get through them. The question is, in which direction will it move? And that's from high to low concentration. Which direction is that? It's from inside the cells to outside the cells. What happens when water leaves the cells? They shrink. Another type of solution is a hypotonic solution. The prefix hypo means below or less than. A hypotonic solution is a solution that is less concentrated than the cells. If that's the case, it means that water is more concentrated outside the cells. Since water moves from an area of higher concentration to an area of lower concentration, and only water can move because the pores are so small, water is going to move from the outside solution inside the cell, causing the cell to expand and sometimes burst. To recap the last slide, in a hypotonic solution, water moves from outside the cells to inside the cells. When this happens, the cells swell and sometimes burst. Some solutions are isotonic. The prefix iso means the same. An isotonic solution is a solution with the same concentration as the cells. Since the concentration of water is the same inside and outside the cell, water will move equally in both directions, and the size of the cell remains unchanged. To recap, in an isotonic solution, water moves equally in both directions. Because it does, the size of the cell remains unchanged.